I want to just begin by saying it's a real pleasure for me to be visiting the Kingdom of Saudi Arabia. And tonight, of course, I'll be receiving the King Faisal International Prize, which is a huge honor for myself and for my laboratory and for the field in which I work. But I have to say that I am particularly delighted to be speaking here at MOEBA because of the central mission of MOEBA. I think trying to identify young people at a time when they're still developing their interests, identifying sort of talent, nurturing it, and promoting it is probably the greatest investment that Saudi Arabia can make, and it's a huge investment for the future. And originally I was planning on presenting a pure science talk, but over the course of the morning I've decided to actually modify my talk ever so slightly and share with you a little bit about my path that brought me to where I am today. And before I begin, I want to just tell you a story that took place about 2 or 3 years ago. So I'm a professor at Harvard Medical School, and I have 2 laboratories. I have one laboratory at Massachusetts General Hospital, and I have a second laboratory at a place called the Broad Institute. This is an institute that's joint between Harvard and MIT, and it's focused on computation, engineering, applied to genomics and to biology. And I share an office at the second laboratory with a Professor Joel Hirschhorn, who is a pretty famous statistical geneticist in the United States. So he and I share an office, and it's about 5 o'clock in the evening, about 3 years ago. And we're just talking about where we grew up. I grew up in Texas, and he grew up in New York City. And for some reason, the topic arose as to what we did when we were in high school at the age of 14 or 15. And he told me that his real love were high school math competitions. And then I told him my real love at the age of 14 or 15 were also high school science fair competitions. And I told him that at that time, when I was 14 or 15, there was a mathematician that had sort of identified me and took me under his wing and served as my mentor. And I told my office mate, his name is George Berzany. And as soon as I mentioned this name, George Berzany, this other professor said, oh my gosh, George Berzany was my high school mentor as well. Now this didn't make any sense because I grew up in Texas, and I don't know how much of geography, of United States geography, all of you are familiar with, but Texas is in a very different part of the country than New York City. And so during rest of that, between 5 o'clock and 6 o'clock, which you just shared stories about how this professor had taken an interest in me, as well as in his career, and this person was extremely influential to both of us. Now 1 week later, I was playing squash with one of my other professor friends. His name is Sebastian Sung. He's a computational neuroscientist at MIT. And after we're playing squash, I told Professor Sung, this funny thing happened last week. Professor Hirschhorn and I were talking, and as it turns out, we have the same high school mentor. And then he asked me, who was his high school mentor? And I said, his name is Professor George Berzany. And he said, oh my gosh, George Berzany was my high school mentor as well. And so before you knew it, there's probably about 10 of us, all of us that are professors in our 40s or so, at Harvard and MIT and at Princeton. All of us were actually connected through a single mentor in our high school years. Okay? And it's this gentleman, Professor George Berzany, whose passion was to identify talented students around the country, and he would write letters. This is well before the internet. He would write letters to each one of us and engage us in unsolved problems. And he would encourage us to develop new solutions to these problems. And he has impacted literally hundreds, if not thousands of kids over a several decade year period. And this last January, I led the nomination of Professor Berzani for the Mathematical Association of America's top prize. It's called the Gungan Hu Prize, and it's the top service award given by the largest mathematical association in the United States. And so he actually received it this last January in Seattle in the United States, and a lot of his former trainees actually went there to sort of honor him. And so the reason I'm so happy to be here today is because I think there's nothing more important than trying to impact students early at the high school level, even if earlier, because once you taste what discovery is like, it's addictive, and you will seek it over and over again. So I think what you're doing over here is really, really amazing. And so I'll use this as an introduction to maybe describe to you a little bit about the path that brought me to where I am today. So when I was in high school, I loved playing tennis, and as a first-year student in high school, we were required by the school to do high school science fair competitions. And so I was a little bit lazy, so I decided to do a mathematics project because all it requires is some pencil and some paper. So I thought that this would be the easiest way for me to complete my assignment without too much work. But as it turns out, I did pretty well, and I won the local high school competition. I went on to the regional science fair competition, and that's where George Berzani read about me. And he wrote me a letter, and it was a bit of an alarming letter because what he said was, "Congratulations on winning the Houston Science Fair. You should know that what you thought you discovered was actually discovered 500 years ago. If you really want to discover something new, why don't you work with me? I look forward to hearing from you." And so on the one hand, I was delighted that this esteemed professor had written to me. On the other hand, I was terrified because he told me that what I thought I had discovered was already known 500 years ago. So I wrote a letter back to George Berzani saying, "Can you please introduce me to some new problems?" And so over my high school time, he introduced me to unsolved problems in mathematics. I would work on these problems over my high school period. I got addicted to mathematics. I even published a few papers as a student in Texas. Okay? So this is where I was in high school, and when I was graduating from high school, I was deciding where to go for college. And I was really fortunate. I got into Harvard, into MIT, into Stanford, into the University of Texas, which is close to where I grew up. But the two things that I loved at that time were math and tennis. And so the decision was relatively easy. I ended up going out to the West Coast to Stanford University. And during this time, during my 4 years at Stanford, I delved deep into pure mathematics. I also fell in love with computer science. This was still early in the era of computers. And I fell in love with the idea of actually using computers to make new discoveries in number theory. But the more mathematics I did and the more tennis I did at Stanford, two things became very clear to me. I am not as good a tennis player as I thought I was. For those of you that know about Stanford, it's probably the #1 tennis team every year. This is where John McEnroe went and played tennis, and so I did not stand a chance against some of the other tennis players at a place like Stanford. What I also realized was that I'm good at mathematics, but probably not gifted in the way that some of the real mathematical giants are. But I still loved it. And this is where another mentor became really important. As a college student, this professor, Samuel Carlin, he took an interest in me, and he actually told me that there was a lot of exciting things that were happening in biology. There were a lot of unsolved problems in biology, and on top of it, biology was, at that time even, becoming an information science. And he told me that there was going to be a need in the future for individuals that could speak biology as well as mathematics. And so as a college student, I worked in the late Sam Carlin's group at Stanford, developing statistical and computational methods for DNA sequence analysis. Um, so that's where I was in college. And then towards the end of college, I had to decide what I wanted to do. So in the United States, after you finish your high school, you do a college degree for about 4 years. Then after that, you go on, if you want to pursue a master's degree, a PhD degree, or a medical degree. And at the time, Wall Street was actually recruiting me quite a bit because people that knew how to do mathematics and computers could make a lot of money on Wall Street. So I came close to going there. But in the end, I decided to pursue a medical degree at the Harvard-MIT program. Okay? And so I shifted from Stanford to the East Coast. And at that time, there was a special educational program, joint between Harvard and MIT, that was designed to train doctors in basic science. And so it's a very physics, chemistry, mathematics heavy curriculum designed to create a new breed of physicians that could do research in chemistry, physics, and mathematics. And so I don't know how many of you have been to Boston or to other cold places, but when I first moved to Boston, it was really beautiful. The very first snowfall is just gorgeous. You know, the trees are beautiful. There's a light coating of snow. I was quite enchanted. And then after a few months in Boston, this is how life felt. Medical school involves a lot of memorization. I see some folks here in the audience that look like they're in medical school based on the scrubs that they're wearing. There's a lot of memorization. I'm genetically optimized for the sun, and I grew up in Texas and went to college in California. So moving to the East Coast was a bit of an environmental shock for me. And life was actually a little bit tough for me at that time. I wasn't sure if I had made the right decision by going to medical school. And then during one of the medical school classes, I was introduced to the topic of mitochondria. Now, this is actually the topic that I work on even today. Mitochondria are actually shown in this image on the upper left. I think many of you know what mitochondria are. These are structures that are found within all of our body's cells, and they're responsible for making energy. They convert all of the food that we eat into biological currency, and they basically power our brains, our muscle, our hearts, so that they can do their jobs. And right around at that time, just a few years earlier, other researchers had demonstrated that the DNA that's found within mitochondria can be mutated to cause a variety of human diseases. And so I'm not exactly sure why, but during that particular cold day in Boston, I was just absolutely captivated by the appearance of mitochondria, the chemistry that takes place within mitochondria, and just thought that this could be an interesting and fascinating topic for me. So for whatever reason, I just got hooked on this topic at that time. And I thought that it could be something that I could specialize in the future. And so in America, we have what are called MD as well as MD PhD programs. And these are different training paths. And so I was trying to decide whether I would do a full PhD or whether it would be sufficient to only do one degree. And I ended up choosing to only do one degree with the long-term goal of actually doing research. And I left Harvard at that time against the advice of all of my advisors. They all said, "You're at Harvard. Why would you possibly want to leave Harvard to go do research elsewhere? That's crazy," they told me. And I told them it's because I found a topic that I absolutely love. There aren't any experts at Harvard in mitochondria, so let me go someplace where I can actually learn about the topic that I'm excited about. So I went to the National Institutes of Health. This is based in the Washington, D.C. area, and this is one of the largest biomedical research campuses in the world. And there was a researcher at that time named Robert Balaban who specialized in mitochondria. And so I ended up working with him at that time to learn the very basics of how energy, it's transformed within our body cells. And then after doing my research medical school thesis at the National Institutes of Health, I came back to then complete my medical school degree. And then at that time, I was faced with yet another decision. And if you haven't gathered this already from my talk, throughout one's sort of journey in academia, every couple of years you get faced with some sort of a decision. Should I do research in Saudi Arabia, or should I go overseas and go do research? Should I study medicine, or should I study engineering? So at every single juncture, there's going to be an important decision to be made. And I tend to tear myself up over every single one of these decisions. I used to spend a few weeks or even a month or two trying to make these decisions, but then, I'm always happy with the decision that I made. And so as I was finishing medical school, I was trying to decide whether to terminate my training in medicine and go do a traditional postdoctoral fellowship, a basic science postdoctoral fellowship, or whether I should do some clinical training. And I actually decided to go ahead and do a little bit of clinical training. And it's because of this, yet another mentor that entered into my life. This person's name is Marshall Wolf, and he was the clinical director of the internal medicine program at Boston's Brigham and Women's Hospital. And he has trained over his career hundreds of now chairmen of medicine at various institutions around the United States. He's a leader in academic medicine. And he really took an interest in me and in my career. And he said, why don't you do a couple of years of clinical training in medicine? This way you can also get exposed to some of the human diseases that are associated with mitochondria as well. So that's what I did. I trained here. And I also did a little bit of work on programmed cell death. Right at that time, the late Stan Korsmeyer had moved to Boston, and he had shown that a process called programmed cell death, or apoptosis, is very fundamental for cell biology and for cancer, and it is largely controlled by the activity of mitochondria. So I ended up working with him while completing my clinical training. And so then after my clinical training, I decided to jump ship and then go back to basic research. And one of the problems that became very clear by seeing patients with mitochondrial disease, so there's a large collection of human diseases, okay, where the mitochondria, right, are malfunctioning. And these are terrible disorders, and I'll tell you a little bit more about them when I describe some of my research. These are terrible disorders. However, at that time, much of the field of mitochondrial medicine was focused on this. It was focused on the mitochondrial DNA. Okay? Now, mitochondria have their own genome that's passed from mother to child, mother to child. Okay? And so most of the textbooks would have you believe that all of these disorders are due to a faulty mitochondrial DNA. As it turns out, only a small fraction of all of these disorders are actually due to problems in this genome. It's just that this genome was well studied and was clearly associated with mitochondria, so it's easy to attribute to some of these problems. But in actuality, most of these disorders had nothing to do with this mitochondrial genome. And that is, of course, because this mitochondria is encoded both by components from the mitochondrial DNA as well as from rest of the genome. And so this first genome was sequenced in 1981. Okay, this was actually the first human genome to be sequenced. This was in 1981, and this was completed by a collaborative team that included Fred Sanger. So Fred Sanger won 2 Nobel Prizes. He's from England. He won 2 Nobel Prizes for the fundamental work that he did both in DNA sequencing as well as in protein sequencing. And thanks to this seminal paper, we knew everything that it encoded. We knew that this genome, this DNA that's found within mitochondria, it encodes 13 proteins. Okay? However, if you take all of the patients that I was seeing at that time, others have seen, a very small fraction of these patients that have faulty mitochondria actually have genetic lesions in the mitochondrial DNA. And that's because rest of the genome is very important for making rest of the cell as well as for making mitochondria. And this genome was coming online in 2001. That's when the draft sequence, the 3 billion bases, the code of life, was coming online. And that was the very year that I was completing my clinical training. And so I thought that this would be exciting now to try to enter into the world of genomics. And so much of the work that I've done over the last 15 years or so has been aimed at figuring out what are all the components that are made by rest of the human genome, the 3 billion bases, that are important for manufacturing our mitochondria. Okay. And so for this, at the beginning, I trained, oh, go ahead, question. Yeah, absolutely. So I'm actually going to jump a little bit into this beginning portion of the talk is a little bit about my sort of biographical path, if you will. And then I'm going to talk a little bit more about the disorders. But for example, there are some familial lactic acidosis, for example. There is a disease called Lee syndrome. I don't know if you're familiar with it, but it's a subacute degeneration of the brainstem. And there's about 75 different genes that can underlie this type of a syndrome. So there's a number of different syndromes that are characterized by cardiomyopathy, gray or white matter disease, blindness, deafness, lactic acidosis. These are some of the manifestations of mitochondrial disease. And I'll get back to those in a few minutes. So in 2001, one of the leaders of the Human Genome Sequencing Project is Eric Lander, who was a professor at the Whitehead Institute at MIT. So I joined his laboratory to train in computational genomics. And actually, that was a very good question that you just asked, because I can tell you about the very first mitochondrial disease that I worked on. And so at that time, there was one disease that was very common in French Quebec. In fact, in this part of French Quebec, this is the most common genetic disease. It's called the French-Canadian variant of Lee syndrome. Okay? And the carrier rate for this disease is about 1 in 20 or so, and it was impacting approximately 1 in 2,000 live births. And these children are born with a dysmorphic facies, so they don't look completely normal. And what they suffer from is a very terrible, rapid degeneration of the brainstem or the basal ganglia, which is the base of the cerebrum. They have lactic acidosis, and they usually die within the first couple of months of life. So this is sort of a prototypical mitochondrial disorder, and you can sequence the mitochondrial genome and it'll be totally fine in this particular instance. This is not a disorder of the mitochondrial DNA. It's inherited in an autosomal recessive manner. And so at the time, there was somebody named Pierre Lavoie who had lost 2 children to this particular disease. And after he lost 2 of his children to this terrible, terrible disease, he decided to do something about it. So he went. He's an amazing athlete. I don't know if you know what a triathlon is. It's when you do a mix of swimming, biking, and running. So Pierre Lavoie at one point was one of the world's top 10 triathletes in the world. And after he had lost 2 of his kids, he decided to do something about it. So he had a bike-a-thon, and he raised $50,000 US that he would then donate for research. And I ended up receiving half of that with the goal of trying to find the gene that causes this terrible disease. And what I did is I wrote a computer program at that time that would basically mine all of the freely available genomic data that was on the World Wide Web. And then I ran the computer program over one weekend. This was approximately in the year 2001 or 2002. And we immediately identified one gene that appeared to be the most likely culprit. And so for those of you that are scientists in the audience, what we did is previous groups had done a technique called homozygosity mapping and identified an interval on chromosome 2 that must statistically harbor the disease gene. In the public domain, even by then, were initial atlases of RNA expression in the form of microarrays and even some initial atlases of protein expression. So these are proteomic data sets. And so I wrote code that would take the genetic interval identified through the human genetics and then asked, are there any other open reading frames within that interval that might be encoding transcripts or proteins that are likely to be mitochondrial in nature? And this led to the identification of a gene called LRPPRC, which then ended up being, we're able to show that this was indeed the causative gene for this disease. And this ended up being a pretty big deal in French Quebec at that time. The government implemented what's called carrier testing so that the transmission of this disease could now be prevented through the use of in vitro fertilization and genetic diagnosis. But it also made a lot of news because of the method that was used. This was almost purely a computational approach that was used to discover this disease gene. And so that was sort of my introduction to my first mitochondrial disease. And then I started my lab at the end of 2003 and 2004. And I started my laboratory already with a real passion and interest for mitochondria. And so just a little reminder that this is what mitochondria look like. This is a single cell. This is what the nucleus, this is the region of the nucleus, but you can't see it. And instead, what are lighting up in this particular cell are mitochondria. Each one of these threads is one of these organelles. And on electron microscopy, this is what it looks like. This is about half a micron in length. It has a double membrane. And within mitochondria is the system called the oxidative phosphorylation system. This is where energy in the form of redox energy is converted into ATP. And mitochondria are quite famous because they are the major supplier of energy for the entire cell and body. And so to some extent, folks in Saudi Arabia should be able to relate to this because Saudi Arabia, of course, is one of the leading producers of oil for the rest of the world. So in a similar way, mitochondria are extremely important for biological energy. And of course, one of the curious features of mitochondria is that they have their own DNA, which looks a lot like bacterial DNA. And so a bit more about these disorders. So when I started the laboratory, we decided that half of the lab would be focused on the basic biology of mitochondria, and the other half would be dedicated to a large collection of what are called the orphan mitochondrial disorders. These are rare genetic diseases. In fact, within Saudi Arabia, there are quite a few rare genetic diseases because of the high levels of consanguinity. These are devastating disorders. Most of these are Mendelian diseases. Most of them are recessive, autosomal recessive diseases. And they can present with any of the symptoms that are shown over here. Because mitochondria are so important for energy production, when they're malfunctioning, they can lead to weak muscles, they can lead to seizures, blindness, deafness. The throat may not work properly, so these patients may not be able to swallow properly. They can develop cardiomyopathies, kidney failure, acidosis. So some of these children typically will present with all of these symptoms. Others will only present with, say, one symptom. And we don't fully understand why some mitochondrial disorders are tissue specific and why others are so ubiquitous in their presentation. There's a lot of excitement in the scientific world and the pharmaceutical world in mitochondria because it's now clear that as all of us age, even if you don't have any obvious disease, as all of us age, the activity of mitochondria declines. And there are some common diseases that now have real links to mitochondrial dysfunction, things like Parkinson's disease, certain cancers, and diabetes, these are all genetically linked to mitochondria. However, some of these common diseases are so complex in terms of their genetics and their physiology, it's very difficult to figure out what's wrong. And so studying these rare diseases where we know the single gene that's defective raises the hope and promise that perhaps they'll provide insights that'll be valuable for not only caring for these rare diseases, but for the common as well. And so what have we done in our laboratory over the last dozen or so years? When I was growing up in Texas, my brothers and I loved cars, and we used to go and buy old cars. We would tinker around with them and fix them, and then we'd sell them, and then we'd buy another car. And the very first thing that we would do when we'd buy an old car is we would go to a store called AutoZone. And at AutoZone, if you tell the person behind the cash register the year, the make, the type of vehicle you have, he will typically then go back and pull out a Chilton manual. And each Chilton manual is specialized for your car. So I would tell him, I have a 1965 six-cylinder Mustang. He would go back, pull out the Chilton manual for the 1965 six-cylinder Mustang. And then when you open these books, right, at the beginning, they literally lay out all of the parts of your Mustang. And then if you keep flipping, these books have wiring diagrams for the air conditioner, for the electrical system, for the motor. And so what we wanted to do in our laboratory is basically build something like a Chilton manual, not for a car, but for mitochondria. And this is what we've been trying to do for the last, decade or so. We have some very simple goals. This mitochondria is super important, it's beautiful, but first of all, we need to identify all of its parts. Right? The sequencing of that tiny DNA by Fred Sanger revealed 13 proteins, but what are all of the other components made by rest of the genome that make mitochondria? So what are the parts? And once we have all the parts, can we connect them? Can we build a wiring diagram that relates these parts to each other? And then once we know what the wiring diagram looks like, can we figure out which of these circuits are important for a disease? And if we have a good understanding of these circuits, can we somehow make an impact on human disease? And so this is basically what we've been doing for the last couple of years. And I just introduced these rare mitochondrial disorders. The hope has been that if we can figure out all of the proteins manufactured by rest of the genome, perhaps that would provide us with a molecular framework for studying these disorders. And we used a technique called mass spec proteomics, so mass spectrometry proteomics, in which we can isolate mitochondria from organs. We can do this from human organs, human cells, animal organs, animal tissues. And we solubilize all the proteins. And then after we solubilize all of the proteins, we chop them up using proteases. So we convert mitochondria into protein mixtures, into peptide mixtures, and then we shoot all of these peptides into a mass spectrometer. And what a mass spectrometer does is it will compute the mass of every single one of these peptides. And if you use tandem mass spectrometry, you go one step further. You actually bombard each peptide, and you get what's called a peptide fragment spectrum. And so from here, we have lots of information about what the composition of mitochondria is. But in order to actually figure out what those proteins are, we also have to take the genomes, for whatever organism, in this case mouse, that we did the proteomic experiment on. And then we take the DNA sequences, and then we translate them in all 6 reading frames to create a theoretical, a library of theoretical spectra. And then what we do is we ask which of the spectra that we actually observed match up to what's theoretically possible. And in this way, we can identify the proteins that must have been in our original mixture. So a lot of computation, a lot of experiments. But in this way, what we've been able to do over more than a decade now is we've been able to identify 1,100 proteins that are made by the nuclear genome that make up mitochondria. So the human genome encodes 22,000 proteins total, and out of those 22,000 proteins, 1,100 of these make up our mitochondria. So that's our parts list, if you will. And so a parts list is just a list, and so what can we do with it? So here's another example of a disease. Okay, this is actually a disease that we saw at Mass General Hospital. This is actually the case of two siblings. They're actually from the Middle East. The parents are first cousins, and then this is a little girl whose care we were involved with, and she had a subacute degeneration of the brainstem. She has lactic acidosis, and if you take some of the biopsy material, we can actually measure her mitochondria, and they're not functioning properly. Okay? And then there's two siblings in this family that are both sick. And so what we've been able to do is we've been able to use the techniques of human genetics to find regions of the genome that must statistically harbor the disease gene. And so these are techniques that were pioneered by Jim Gusella, who's a former King Faisal Prize recipient. And so we use those types of methods to find a region of the genome that must harbor the disease gene. But then what we can do is we can ask which of the genes in this interval are also encoding proteins that find their way to mitochondria. And then in the end, in this intersection, there's very few proteins that emerge. We can sequence them, and then we can find the disease gene. And this is a bit of a, I need to update this particular slide, but over the years, we've now discovered about 15 to 20 different genes, which when broken, give rise to these terrible diseases simply by using this parts list. Okay. So I'm gonna, probably the most important slide that I can show is a slide of my team. Over the years, I've been very fortunate to be able to recruit extremely talented students and fellows and physicians and technicians, men, women, from all different ethnicities. And we've all collaborated together on our mission to try to decipher mitochondria, its physiology and its role in disease. And I want to end this portion of the talk returning to my path, because today in the era of genomics, it's quite clear that if you're well skilled in mathematics and in computation, there is a special role for you in this particular science. So a lot of people, a lot of my students say, hey, that was really forward-looking of you. You majored in math, you went to medicine, you trained in genomics. That was such a very linear path. But my path is only linear in retrospect. Right? What I want to emphasize is that at every given stage, right, and this is sort of a message that I want to try to impress upon some of the students and younger people here, at every given stage, whether it was at the end of high school, at the end of college, or at the end of medical school, I was faced with some sort of a decision. And had I gone down this path, maybe I'd be on Wall Street right now making a ton of money, right? And maybe that would have been a fine life. It would have been a different life. Maybe I would have been a private practicing cardiologist in Texas. That would have been a fine life. And this is the path that I took. And so in retrospect, it's very linear, but at every given stage, there's so many different options. And living life really means making a decision, moving forward with it, making the best decision possible given the information that you have at the time, and then just knowing for yourself that everything is going to be okay. And to the extent that you can, I would urge all of the young people here to always follow their heart. Try to do whatever really excites you, because at each stage, you're going to end up accumulating experiences as a student, as a fellow, as a postdoc, whatever you do. And it's that mix that makes you so unique that will allow you to make discoveries that nobody else could even imagine. And then I also want to just emphasize that my professional career would have been very boring and probably very unsuccessful were it not for the fact that I had a number of different mentors beginning in high school. Even to today, I have mentors that really help me and counsel me in any of the questions that I have. And I would urge the young people here today to actively seek out mentorship, because older people love talking about themselves and advising and trying to guide younger people. So you have to actively seek out mentorship. It's not going to come through some program that matches you with a mentor. You need to go and find somebody with whom you can relate. And what I have to say as a professor is I also love just mentoring as well. A number of my former students now have gone on to lead their own research groups, and I'm just highlighting a few of them. And they've made some of their own discoveries in their own laboratories around the world, major discoveries that have nothing to do with me at this point. And it gives me as much joy to see them excelling as it does when we make our own discoveries in our laboratory. And so that's all I wanted to say in terms of my path. And depending on the amount of time and interest, I could go a little bit deeper into one of the newer science projects, or I can open it up for questions. No, it's a great and actually it's a very timely question as well. So it's like, again, this was not a particularly deep science or medicine talk. It's a bit of an overview talk, but let me address your question from a bit more of a scientific and medical perspective. And so a small fraction of these disorders are due to mutations in the mitochondrial DNA. Most of them are Mendelian and due to mutations in the nuclear genome. And so I think for the Mendelian disorders, I think there's a path forward in countries and in societies where pre-implantation genetic diagnosis and in vitro fertilization are possible. I think there's no reason why we can't prevent the transmission of those disorders for future generations. And so the first example I gave from French Quebec, that particular disease, because of a founder effect, not that different than high rates of consanguinity, that disease is very common. And so the government, because they have supported genetic screening, and they reimburse for things like pre-implantation genetic diagnosis and IVF, that disease that I described back in 2002, 2003, it's almost been eradicated at this point. There's only 10 children now with that particular disease, and many of those parents now have healthy children. So I think that for the Mendelian disorders, there's a path forward. Now, for the 15% or so that are due to mutations in the mitochondrial DNA, you're raising a very, very important idea, one that's now being considered throughout the world. There's a procedure called mitochondrial replacement technique, or MRT, where, and just for the purpose of the others in the audience, what's done is an egg from a woman that has mitochondrial DNA disease, her nucleus is removed, and then there's a donor egg, the nucleus is removed, and then the intending mother's nucleus is then injected into the donor oocyte. It's fertilized, and now you have an embryo that's constructed from the DNA of 3 different parents. This can now be implanted in the mother, and it can actually give rise, in theory, to a healthy child that doesn't have the mitochondrial DNA mutations. So this is a technique called MRT. It's a radically new approach. It appears to be working in non-human primates. This has been proposed in the United Kingdom. It's received approval to move forward in a clinical trial phase in the UK, effective January 2016. And this has been brought forth to the United States Food and Drug Administration as well. So I was actually on the committee, on the bioethics panel that reviewed the bioethics of this procedure for the last year, because this raises all sorts of bioethics questions about whether this is ethical. Are we editing the germ line? Should we be crossing this line? And in fact, our report, there's a press release for our report that's taking place today in the United States, but I'm here, of course, and so I can't be there for it. And so our committee actually said that it is ethically permissible to move forward with that particular procedure in a clinical trials setting, if initially it's limited to male embryos so that it cannot be passed on to future generations. And so I think what you're raising is really at the cutting edge of sort of genetic technology, reproductive technology. It's beginning to take place in the UK, possibly elsewhere in the world, but this is something that we need to sort of move forward with in a clinical trial setting with incredible care and caution, because it is involving humans and it is experimental. No, I think this is actually an area, as you know, Wafa, that Saudi Arabia can actually be a world leader because many of these genetic disorders are pretty common. These are rare disorders, but comparatively common in Saudi Arabia and the Middle East. So this is a place where you can be world's leaders. I was born in India, and then we immigrated to the United States when I was about 2 years old, when I was very young. And my father is a retired surgeon. He trained in the United States, and then he practiced in Texas for about 35 years or so. And I have 3 older siblings that are also doctors, and so the big joke was that we carry the medical school gene in our genome. So it's a family of doctors. And my parents have always been incredibly supportive of anything that I wanted to do. Whether I wanted to go into mathematics or whether I wanted to go into medicine, they've just been incredibly supportive. And I think in order to really excel at a young age, you need to have that family support as well. If you're having to struggle with issues at home or aren't comfortable on the home front, there's no way that you can do original research. So nothing that I've done would be possible were it not for my parents. You know, we actually had a few of these discussions earlier, and all I can say is that professors, especially at research institutions, most professors have way too many ideas, and at the end of the day, we can only pursue a few of them because it takes a really talented student or fellow to actually run with the idea. And so I think there's actually a huge, if there are talented students here, there's no reason we can't try to formalize some sort of a program between Harvard or MIT or another place that connects professors with ideas, with students that are excited about learning how to do science. I do think that Harvard is a special place, and that's, I mean, I'm there because it's a special place, but I think what you're describing is also just, it's the value of having high quality mentorship, right? So the professor, George Berzany, that impacted me, my office mate at Harvard, my squash partner who's a professor at MIT, he was not at Harvard. He was at a relatively small school in the United States. But what he did was when he found a student that was pretty intelligent and very hardworking, he introduced to that student unsolved problems. He instilled in that student the importance of taste, right? Not all problems are equally worthwhile pursuing. There's good problems to pursue, and there's ugly problems to pursue. He instilled in us sort of the importance of aesthetic as well, and the questions that one pursues. And so I think what you're describing is sort of the incredible importance of having high quality mentors that can guide very smart and talented people, because that way, that's what lights the spark in a young person. And that spark can come from a Harvard professor, but it can come from almost anyone else that's just a bit more experienced. I think this is a great question. So whenever there's a new experimental therapy, we never know in advance whether the good will outweigh the bad. There's always a balance between risk and benefit. And what our ethics committee reviewed was not the science or the clinical medicine. We didn't review sort of the scientific safety package. That's the job of the Food and Drug Agency in the United States. What our committee focused on was the bioethics. Is it ethically permissible to move forward in such trials in a research setting, just to be clear? From a scientific perspective, I think there's work from the model organism world suggesting that if two organisms are extremely, if two members of the same species are extremely diverged, there might be what's called mitochondrial nuclear incompatibility, which could lead to some detrimental consequences in the offspring. However, as far as we know, within humans, there hasn't been such divergence that there would be such incompatibilities. And so as in many other sort of research endeavors, we simply don't know. It's not until additional research is done that we'll know whether or not this could be a safe or unsafe procedure. And even things like in vitro fertilization at that time, that actually wasn't pursued in sort of a controlled research setting, folks started applying in vitro fertilization, and it turned out to be a relatively safe procedure, but it's something that we can say is relatively safe only in retrospect after having a lot of experience. And so I think it's still it's still an early time in the history of this technique, and we have to proceed forward with extreme caution, and only in a very, very circumscribed set of cases where it's clear that the alternative is to have a very, very unhealthy child. Our committee, just to be clear, we didn't approve the technique. That is not what this committee did. What the committee did is it reviewed the bioethics of moving forward with clinical trials, and the FDA in the United States is responsible for reviewing things like safety, and of course, only if all of the preclinical data, potentially some even human studies that are investigational, only if they deem that this is a safe procedure would it be possible to move forward. So just to be clear, we reviewed sort of the bioethics of the procedure as opposed to the safety of the procedure. And in any ethics calculation, the benefits have to outweigh the risks. And so if it looks like it's a risky procedure from a safety perspective, I doubt that the FDA would actually approve it at that point. I mean, sometimes it just requires remarkable persistence. I trained in a field called mass spectrometry proteomics. And at that time, the world's leader was in Denmark. And so I wrote an email to him asking if I could work with him. He never dignified my email with a response. And then I emailed him again. He never wrote back. Then I saw that he was giving a talk in Boston. So then I emailed him again saying, can I meet you? He never responded. So then I wrote 4 more emails saying, I'm planning on coming to your talk. Can we meet afterwards? He never responded. And then I went to his talk. I was there 15 minutes before his presentation. I went up to him, and before I could say anything, he said, you must be Vamsi Mootha. And I said, yes, I am. And then he said, let's talk after my presentation. And I had a talk with him afterwards. And eventually he invited me to come and work with him in Denmark. And I worked with him. I didn't go through my entire history, but I spent about 6 or 7 months with him towards the end of my clinical training, working with him in mass spectrometry. He's one of the leaders of proteomics today. So I think sometimes you have to, if you know what you want to do, sometimes you just have to be persistent, hopefully in a gentle and polite way. But I think persistence really pays off, because some of the busier scientists, some of the more, quote, famous scientists, they may be so busy, they have good intentions, but your email may just be lost. So be persistent.